All right, folks. I'm going to go ahead and get started. Welcome to our ADA Pathways session for December. Unfortunately, we have originally two speakers scheduled, Tom Delong, who is here, Thomas Delong, and Michael Stitzel, who informed us a couple of hours ago they had something urgent come up and won't be able to attend. So we're going to reschedule Michael for one of the three 2002 sessions that we have lined up. So right now we're going to have January, February, and March sessions. We hope to put Michael Stitzel in the February session. And Kylie is going to be sending out the schedule of talks once we have firmed up Michael's participation, which will be attached to your calendar invites as the 2021 schedules have been. That'll be replaced with the 2022 schedules with 3 sessions planned for 2022. So Thomas DeLong is our presenter today. He's at the University of Colorado Health Sciences Center in Denver. His type of award was the Accelerator Award, which began in 2015. And he's going to tell us today about hybrid insulin peptides. How do they form in beta cells? Thomas.
All right, thank you very much. All right, I'm Thomas, and I'm talking about hybrid peptides and how they form in beta cells. But I'll also give you some background to tell you what hybrid insulin peptides are and so on. So in my lab, we study type 1 diabetes, and the animal model that we use to study type 1 diabetes is the non-obese diabetic or NOD mouse. And about 80% of the female NOD mice develop autoimmune diabetes within 1 year. In type 1 diabetes, CD4 T cells play a key role in the destruction of the insulin-producing beta cells. So here's a picture of a CD4 T cell, and unlike an antibody, a CD4 T cell does not directly recognize its target antigen. Instead, a fragment of the antigen, a peptide, has to present it in context of the major histocompatibility complex MHC class II on the surface of an antigen presenting cell. Only then will a CD4 T cell recognize its target epitope and become activated. Now this slide here shows how we envision the role of CD4 T cell in the destruction of the insulin-producing beta cell. Now here's here pancreas and the pancreatic islets. And initially an antigen presenting cell picks up antigen in the pancreas, migrates to the pancreatic lymph node, where it presents a peptide fragment in context of MHC class II to the autoreactive CD4 T cell. The CD4 T cell then migrates back into the pancreas, where it encounters more antigen and starts to proliferate and produce cytokines and chemokines. This leads to the recruitment of other effector cells, such as macrophages and B cells, which then initiate the destruction of the insulin-producing beta cell. After the damage is done, most of the CD4 T cells die. However, some of them make it to the periphery, where they form a CD4 memory T cell population. Now, my mentor, Katie Haskins, here at the University of Colorado, had in the past isolated CD4 T cell clones from diabetic NOD mice. Here's a list of her T cell clones, and these T cell clones are all unique, and they can all trigger diabetes in NOD mice. I've spent more than a decade trying to figure out what the antigens are that are recognized by the T cell clones. We found, for example, one T cell clone responds to a natural cleavage product of islet amyloid polypeptide called amylin. These T cell clones respond to a hybrid insulin peptide. I will talk about hybrid insulin peptides in the next slide. There are 3 more T cell clones. They're all unique. They all have a distinct T cell receptor, and they all trigger diabetes, target a different hybrid insulin peptide. And then there are several T cell clones that respond to the insulin B chain, in particular the B chain epitope B9 to 23. And we think that the natural antigen may actually also be a hybrid insulin peptide, or more than one hybrid insulin peptide that's recognized by these T cell clones. Now, what are hybrid peptides? Now, hybrid insulin peptides form through covalent linkage of an insulin fragment to another peptide fragment from different proteins, such as chromogranin A, IPP, or even insulin. You can see that the resulting amino acid sequence contain a non-genomic amino acid sequence, making them a plausible target for autoreactive T cells, because they would look foreign to the adaptive immune system. Now here you see a picture of an insulin-producing beta cell. You see the nucleus. The little black dots here are the insulin granules. And when we enrich granules from the insulin-producing beta cells with differential centrifugation, the T cell clones from Dr. Haskins' panel respond to these insulin granules, to the insulin content in context of an antigen-presenting cell. So what I did in the past, we used beta cell tumors from a transgenic mouse that contain lots of antigen that's recognized by the T cells. And we broke open these beta cell tumors, and through high speed and low speed centrifugation steps, we enriched a pellet that was highly enriched in insulin secretory granules. These granules contain the antigen recognized by the T cell clones. We then lysed these granulolysates and ran them over a size exclusion column with the overall aim to identify the antigen targets by mass spectrometry. Now what we see here is a size exclusion chromatogram. The blue line is the proteins eluting from the chromatography column over time. And then we took fractions that we eluded and used the T cell clone BDC2.5 to determine which fractions contained the antigen, which was measured by the interferon gamma T cell response. You can see here there was a small set of fractions that the T cell clone responded to, telling us where the antigen was localized. Now in 2010, we identified a protein, chromogranin A, and a natural cleavage product of the protein, chromogranin A, that was recognized by the T cell clone. However, when we looked at these fractions by mass spectrometry, looking for this natural cleavage product that's called W14, you can see that it did not follow the distribution profile that was given by the T cell clone BDC2.5. So the T cell clone responds to the natural antigen. However, the peptide W14, which the T cell clone responds to, did not perfectly coelute to this peptide. However, when we looked at the insulin C peptide and peptide fragments in these fractions, we could see that insulin C peptide perfectly coeluted with the natural antigen in these fractions, indicating that C peptide may be the antigen for the T cell clone. We obtained different concentrations of C peptide and assayed them with the T cell clone BDC2.5 to an interferon gamma ELISA assay. And you can see when we used high concentrations of C peptide, shown in orange here, the T cell clone would not respond. However, the T cell clone gave a weak response to very high concentrations of a C peptide fragment, shown in green here. And it did respond to the peptide W14 of chromogranin A, shown in blue here, but only at high concentrations. However, when we cross-link the green and the blue peptides, making a hybrid peptide, you can see that the T cell clone responded to this peptide to very low concentrations. We then went back to look at our granulolysates to see if the hybrid peptide was in there. And for this, we did a proteolytic digest to cut out the core peptide. We need short peptides for mass spec analysis. And we're able to find this peptide in the fractions. So you can see here is the mass spectrometric fragmentation spectrum of the peptide that we found in the granulolysates. And here's a synthetic peptide that we obtained to compare if the synthetic peptide's fragmentation spectrum looks like the natural peptide, when you can see they look almost identical, giving us a high degree of confidence that these peptides are actually in the granulolysates. We've since spent significant time on trying to show that hybrid insulin peptides are present in islets of mice and in islets of non-diabetic human organ donors. And we found several hybrid insulin peptides. Here are the HIPs that we found in mice by mass spectrometry with high confidence. For example, we found a whole list of C-peptide fragments that were linked to various natural cleavage products, such as C-peptide, an IEPP peptide, or a chromogranin A peptide. And we found them with very high confidence in mouse islets and also some of them in human islets. Now, you can see here 3 T cell clones from Dr. Haskins' panel responded to this hybrid insulin peptide between the C-peptide fragment and the peptide W14 of chromogranin A. 2 T cell clones responded to a hybrid insulin peptide between the same C-peptide fragment and a natural cleavage product of islet amyloid polypeptide. And then here is another T cell clone from a different group from Pierre Santa Maria in Canada that also responded to a hybrid insulin peptide and also triggers diabetes in NOD mice between the same C-peptide fragment and the N-terminus of C-peptide. This hybrid peptide here, the C-peptide hybrid, we call HIP11. And we also found T cell clones in residual pancreatic islets of organ donors with type 1 diabetes that target this HIP, as well as in the peripheral blood of recent onset type 1 diabetic patients. So when we look at all the HIPs that we so far identified by mass spectrometry in mouse and human islets, and all the T cells that we and others have identified in that play a role in disease, such as in the residual islets of organ donors with diabetes and peripheral blood of type 1 patients, or in NOD mice triggering diabetes, you can see that there's only a small number of HIPs for which we only have mass spec evidence for their presence in beta cells and T cells targeting these peptides. And these are shown here. Now, HIP11, which is the hybrid peptide that forms between C-peptide and C-peptide, we found by ELISpot analysis in recent onset patients, where we were looking at the peripheral blood to determine if there were CD4 T cells in there that responded to this hybrid peptides in the patient. And you can see here in the new onset patients, we had a large number of patients that responded to HIP11, so by counting the number of spots by ELISpot analysis responding to this peptide, but our negative control in the absence of antigen did not yield a response. Now, when we compare this to the control subjects, which were not diabetic, we only had one outlier here that responded to this hybrid insulin peptide. However, none of the other control subjects responded to this HIP. Now, this outlier here was a first-degree relative of a type 1 patient, so I wouldn't be surprised if we saw that patient show up here at the Barbara Davis Center in Colorado one time sooner or later. Now, the question that I've been focusing on over the last years is, how do HIPs form and where do they come from? And there are several mechanisms of how HIPs can form, and I'm going to talk about the protease-mediated mechanism here today, because I think that's the one that's responsible for making these disease-relevant HIPs. Now, when we go back and look at the table of disease-relevant HIPs, for which we have T cells and mass spec evidence, you can see all of these HIPs contain the same blue left side peptide ending in the amino acid sequence LAL in mice and in humans. We found these peptides with high confidence in the islets of mice and humans, and we found T cells responding to these hybrid peptides by mass spec. Now, the primary function of a protease is actually the cleavage of a peptide bond. And the way a protease does this, here's the insulin C peptide of a mouse. It would attack this peptide bond at the LAL residue, form a reactive intermediate. Now, in the presence of water, this reactive intermediate would react with the water and leading to the cleavage of the peptide bond and the release of the free peptide. This is the primary function of a protease, such as in your stomach, for those who just had lunch. However, in the insulin granules, we have a very low water content, and we do have high contents of other natural cleavage products, such as the peptide W14 or the chromogranin A peptide WQ6, which is shown here, which is the N-terminus of the peptide W14. And in this scenario, in the insulin granules, this peptide can compete with water over this reactive intermediate, thereby driving the formation of a hybrid insulin peptide, shown at the bottom. This would be the hybrid peptide sequence that's recognized by the T cell clone BDC215. So the protease would attack this peptide bond and either cleave it or lead to the formation of a hybrid insulin peptide. Now, when we look at this left side peptide here that forms to the protealytic cleavage, this peptide is called DES2731C peptide, as it lacks the 5C terminal amino acid sequence of C peptide. Now, this peptide cleavage product of C peptide had previously been identified by Dr. Bruce Vercheer in Canada. What Bruce had done, he took rat beta cells and pulse chase labeled them with tritiated leucine. He then took the rat beta cells and exposed them to a high glucose cocktail, causing the insulin granules, the beta cells, to release the insulin granule content. He then looked at the released content to determine how much radioactive insulin he could find in there by fractionating the samples by HPLC. And you can see here are the two insulins. Mice and rat have two insulins, unlike humans, that eluded, which was one of the major secreted products due to the glucose stimulation. the granules also released insulin C-peptide, and you can see a major C-peptide cleavage product that was co-released with insulin and C-peptide was DES2731 C-peptide. Now, this puts the protease that drives the cleavage of the C-peptide, leading to the formation of the DES peptide, into the insulin secretory granules, because the glucose-stimulated insulin secretion only takes place from the insulin granules. So my student Samantha Grafford in my lab, she went ahead trying to purify the protease to identify which protease is responsible for the formation of these hybrid insulin peptides that have this DES2731 C-peptide fragment on the left. And she did the same approach that I had initially followed to identify the hybrid insulin peptides, but she had a different goal. She didn't want to purify the hips. She wanted to purify the protease responsible for making the hips. So she took these beta cell tumors, broke them open, enriched the granules, and fractionated them on a chromatography column. She then took 8 fractions. So she did a different type of chromatography than I had done initially, took these 8 fractions, and these fractions should contain the protease that drives the formation of the hybrid insulin peptides. And then she put in the hybrid peptide precursors. And if the protease is present that makes the hybrid peptide, then it should start forming in the fractions that contain the protease. So initially, she took these fractions without the peptides and incubated them for a couple of hours and assayed them with the T-cell clone BDC2.5, and she could not detect any hybrid insulin peptides. Now, she did not detect hybrid peptides because, first of all, she used very little material. She used far less material than I had initially done. And she incubated the fractions in the absence of protease inhibitors. And if there are proteases in there, the proteases would possibly chop up the hybrid peptides that were there to begin with. However, when she added the hybrid peptide precursors, allowing the proteases in there to drive the formation of hybrid insulin peptides, you can see that we did have hybrid insulin peptide formation as measured by the T-cell response to the resulting HIP. She then went ahead and mass spec'd all these fractions to determine what type of proteases are in there. And you can see we found over 30 different proteases from 5 different families: aspartic, metallose, cysteine, threonine, and serine proteases. Now, we could, of course, have gone ahead and gotten all these proteases to test which of them can make the hybrid insulin peptide. But we decided, let's narrow down the list of hybrid insulin peptides by using protease inhibitors. For example, if we had a protease inhibitor that blocks cysteine proteases, and the cysteine protease was responsible for making the hybrid insulin peptides, then we would not observe hybrid insulin peptide formation. So we repeated these experiments in the presence and absence of protease inhibitors, and this is what we observed. When we used the protease inhibitor E64, which inhibits cysteine proteases, you can see that the hybrid insulin peptide formation went down a little bit, but not significant, not very much, just a tiny bit, especially in fraction 4. Now, when she used another protease inhibitor that inhibits aspartic proteases, this inhibitor is called pepstatin, you can see that we almost completely knocked out the hybrid insulin peptide formation. At least we probably knocked down 80% of the formation. When we used a combination of these protease inhibitors, E64 and pepstatin combined, you can see almost all the HIP formation capacity of these fractions was gone. So this data suggested to us that there are at least 2 proteases in there that make hybrid insulin peptides. One of them, the primary one, is an aspartic protease, and a secondary one is a cysteine protease. Now, luckily, there was only one aspartic protease in our fractions, and that was cathepsin D. We then went ahead and obtained a recombinant form of cathepsin D and repeated the experiments in the presence and absence of cathepsin D by adding the peptides to the reaction mix. And then we used the T-cell clone BDC2.5 to determine how much hybrid insulin peptide would form. We did this reaction at various pHs. In particular, pH 5.5 is about the pH of the insulin secretory granules. pH 4.5 to 5 is the pH of the crenophagic bodies, which may also be a site where these hybrid insulin peptides can form. But you can see at all of the pHs, we had hybrid insulin peptide formation catalyzed in the presence of cathepsin D, but not in the absence. The reaction went best at lower pHs, but it also took place at higher pHs. Now, a major problem that we had throughout these reactions was actually that sometimes we found hybrid formation in the presence of our fractions, sometimes we didn't. And that had to do with the amount of sample we had in each experiment, which varied, and with the incubation time that we used to make these hybrid peptides. So incubation time is a key here, because as you can see here, if we incubate our samples for a short time in the presence of cathepsin D, after 2 hours, we have a maximum of hip formation that we can detect. However, if we incubate the samples even longer, the hybrid insulin peptide disappears again. Now, how can that be explained? Well, we think what's happening here is the hybrid insulin peptide is made, and as the C-peptide is being used up, there's more and more reagent available until eventually all of the C-peptide is actually a hybrid or a chopped up form of C-peptide, and then no more hybrid can form, and the protease starts chopping up the hybrid peptide again. So after long incubation times, the hybrid insulin peptide gets eaten up again, and all the C-peptide will be chopped up. So the message here is the proteases cannot, the cathepsin D cannot only make hybrid insulin peptides, it can also break them. To determine the specificity of cathepsin D, we took mouse C-peptide, shown right here, and incubated it in the presence and absence of various concentrations of cathepsin D. Now, the more cathepsin D we add, the more cleavage we expect to take place at a specific site. For example, when we look at this glutamine residue here, in the absence of cathepsin D, we have no cleavage here, but the more cathepsin D we add, the more cleavage we get. At very high concentration, the yield goes down again because we use the mass spec signal to obtain peptides describing this cleavage site. Now, as you can see here, there seem to be 2 major cleavage sites, which are at the same time also proteolytic transpeptidation sites that the cathepsin D favors. The first one is our LAL site, that's the most dominant one, where we get most of the cleavage. And the second one is after the TL residue. So cathepsin D is really specific at making hybrid peptides at these 2 different sites. And when we look back at our mass spec data, you can see that we have hybrid peptide forming not only at the LAL site, that's where we find most of the hybrid peptides by mass spec, but also after the TL site. There's one hybrid peptide that we identified with high confidence that formed at this location. We also treated human cathepsin D with human C-peptide, and that is shown here. And you can see the human cathepsin D was very specific at cleaving insulin C-peptide at the LAL site. And that leads us to think that the cathepsin D is the primary protease driving the formation of HIP11, to which we found T-cell responses in the peripheral blood of new-onset patients, as well as the residual islets of type 1 diabetic organ donors. We also obtained a human T-cell clone that we had isolated from a new-onset patient. And this T-cell clone targets HIP11, the C-peptide hybrid. And you can see the T-cell clone responded when we treated insulin C-peptide in the presence of cathepsin D, and most of our hybrid insulin peptide formation took place at pH 4. We validated these proteases form hybrid peptides also by mass spec, but I'm only showing you the T-cell assays here. We then also obtained insulin and human insulin to determine whether cathepsin D can also cleave within the insulin sequence. And you can see here, cathepsin D like to cleave in several sites of the insulin A chain and also in the insulin B chain. In particular, the site over here is a primary cleavage and potential transpeptidation site for cathepsin D. I told you earlier, we had several B9 to 23 reactive T-cell clones in Dr. Katie Haskins' panel. And you can see cathepsin D likes to cleave right at the core of insulin B9 to 23. Interestingly, when Dr. Maki Nakayama at the Barbara Davis Center here took NOD mice and mutated that tyrosine residue to an alanine residue, the mice would be protected from diabetes. Now, I like to think that this is because no more hybrid peptides can form, because you can imagine if you mutate the primary target of cathepsin D, the tyrosine here, into an alanine, the cleavage site specificity would go down significantly, and we would no longer lead to the formation of hybrid peptides at that location. I told you also that E64 inhibited a little bit of hybrid insulin peptide formation. So we obtained the list of cysteine proteases that we thought could be relevant for the formation of hybrid insulin peptides in the samples. And we repeated our experiments to compare it with cathepsin D. And you can see here, cathepsin D in black led to the formation of hybrid insulin peptides at all the conditions that we tested, but also the cathepsin L, but only at the lower protease concentrations. And again, I think this is because the hybrid insulin peptide forms at low protease concentrations, but at higher concentrations, all of the insulin C-peptide gets used up and the hybrid peptides gets broken down again. So if we add more protease or if we incubate it for a longer time, the hybrid peptide gets broken down again. Now, cathepsin L had previously been implied by a collaborator in Denver that said cathepsin L is responsible for the formation of hybrid insulin peptides in beta cells. And what he had done, he taken a fragment of insulin C peptide shown right here, and the fragment of the chromogranin A peptide right here, and with the goal to see if cathepsin L can lead to the formation of a hybrid insulin peptide. And he used mass spectrometry to show that this is happening. We used two types of cathepsin L. One of them was recombinant, one of them was purified, I think from liver. And you can see that the hybrid peptide formation of cathepsin L was very specific to the LA residue and to the LE residue, but the specificity to the LAL residue, for which we find hybrid peptides in mouse and human islets, was rather low. So we think that cathepsin L does not have the specificity profile of hybrid insulin peptides that we find by mass spec in human islets. And we don't find any hybrid peptides in mouse and human islets that form at this location or at this location. We only find hybrid peptides at that location. So we obtained islets from a CAT-L deficient mice to see how much hybrid peptide is in these islets. And the results are shown here. So we used the T-cell clone BDC215 and various concentrations of islet cells. You can see in the NOD islets, the T-cell clone would respond to these islets. And as we go down with the islet cell concentration numbers, we have less hybrid peptide. But we can see in the absence of cathepsin L, such as the CAT-L knockout or in the heads, we have even higher hybrid insulin peptide. Now, what we think is happening here, cathepsin L's primary role is the chopping up of peptides, in particular in the lysosomes, even though cathepsin L has also been shown to be in the insulin granules. Now, what we think is happening here, the hybrid insulin peptide recognized by BDC2.5 is made by cathepsin D. And in the absence of cathepsin L, it's no longer chopped up. So more hybrid insulin peptide can form. And that's why we formed more hybrid insulin peptide in the CAT-L knockout islets, because the protease CAT-L is no longer there to chop up the hybrid insulin peptide that forms by cathepsin D. We also did this with another hybrid insulin peptide. The T-cell clone BDC9.3 reacts to another hybrid insulin peptide in mouse islets, and we observe the same pattern here. In the absence of cathepsin L, we have more hybrid insulin peptide present than in the presence of cathepsin L. Now, in summary, I think there's one primary take-home message that I want you to take with you. We think cathepsin D is responsible for the generation of disease-relevant HIPs in beta cells. We think that in the absence of cathepsin L in NOD islets, yields an elevated HIP content, indicating the role of cathepsin L in the degradation of peptides in HIPs. So we don't think cathepsin L plays a critical role in the generation of disease-relevant HIPs. And lastly, proteases not only make, but also break HIPs. Now, this brings me to the end of my talk. I want to thank the people in my lab, in particular Sam. She has done outstanding work purifying that protease. We're just putting the paper together right now and we'll be submitting it soon. My collaborators at the Department of Immunology, in particular Katie Haskins, my mentor here, our collaborators at the Barbara Davis Center, and Hubert Say at the University of Birmingham, who provided us with the CAT-L knockout islets. And with this, I'm happy to take any questions that you may have for me.
Wow, thank you very much. That was a real tour de force of elegant biochemistry. Thank you very much. Questions either can come in the chat, or we could have people just blurt it out if they'd like. Anybody want to ask a question?
This is Lloyd Smith. I'll ask a question. Hello, Thomas. We haven't met before, but I've been watching your work and find it very interesting. So thanks for giving this talk. I was just wondering, in those last few plots that you showed, where you use the ELISA spot assay to quantify the peptide, how come you didn't do that by an SRM assay, something that would be more molecular as opposed to an immunological assay?
To look, you mean the ELISpot assays, or?
Well, your last few slides where you kept showing.
Oh yeah, so we did look at the cathepsin L knockout islets by mass spec, and we did validate by mass spec that hybrid peptides are in there. However, the quantification is better with T cells. I actually also think, for the most part, the T cells are more sensitive than the mass spec instrument that we're using. T cells are amazing. They're really, really specific, and they're super sensitive, and they can pick out very low abundant concentrations of these peptides. I would like to put this this whole story a little bit in context with the recently, just 2 weeks ago, I don't know if you read the New York Times on the weekend, but they have just actually transplanted stem cell derived beta cells into type 1 diabetic patient. And that patient, one of those patients, which they gave the lowest possible dose of islets to test the safety of it, is now free of insulin treatment. Now, what the patient is not free of these days is actually immunosuppressants. And that's the problem with type 1 diabetes. We not only have to replace beta cell, but we also have to keep the immune response in check. And what I like to hypothesize, and we're working on testing that hypothesis, is that cathepsin D is really the primary hybrid peptide antigen forming enzyme in the human and in the mouse system. And I can imagine that we can potentially make stem cell derived beta cells that will lack cathepsin D. And if cathepsin D is no longer there, the target for these hip reactive T cells will also be gone, and the T cells will have nothing to go after. Now, whether that is enough to stop the immune attack on the beta cells, I do not know. There could be problems with epitope spreading, but there could also be that these peptides are primary epitopes that are needed for the to initiate the attack on the beta cells. So I'm really interested in cathepsin D as a therapeutic target, and potentially allowing us to prevent the formation of hybrid peptides and people from getting diabetes. We also looked at the myelin sheath of organ donors with multiple sclerosis, and we also found one protease in there, and that happened to be cathepsin D. I don't want to put everything in the field of cathepsin D, but I think cathepsin D is an interesting target, and I wonder what else we can do with it.
So let me follow up on that. So it's sort of in the vein of what I was thinking. So you've got this elegant evidence that you make more hips when you manipulate cathepsin D, yeah, in rodent islets.
Yeah.
And the problem with trying to determine whether a similar phenomenon exists in human islets in type 1 subjects is, by the time they turn up as type 1 subjects, they essentially have no beta cells, and certainly not beta cells that you can get access to, right? Yeah. So how do you cross that bridge to show that this pathway is relevant in human islets, other than the elegant approach that you just described?
So I do have a collaborator here in Denver who's working on making induced pluripotent stem cell derived beta cell clusters. And we're just starting to test that to see whether we can find hybrid peptides in there. And then we want to use that system to see if we can block it from these beta cells.
So I have a question when I can call on me when I can speak, Chris.
Yeah, you're on, buddy.
All right. Tom, you know I love this work, and I have for a long time. So I have a very practical question first, and then some other ones. But the practical one is, you showed the ELISpot assay to the hybrid protein. Is that being made commercially available? I think the T1D community would be very interested in an easy T cell based assay. So
the ELISpot is not really commercially available. I mean, we did the study on 40 patients and 40 control subjects or so. But I think it was a very, very striking result. Let me just share that with you one more time. It was done at the Barbara Davis Center with Peter Gottlieb. And what we did here is we took, now where is it? We took the new onset patients that had diabetes for less than a year, isolated the peripheral blood, BMCs, and incubated them with and without HIP-11. And
I think it's great.
It's really striking how many type 1s, you can see which ones have a higher response over background.
Yeah.
And that was not the case in the non-diabetic control subjects. So it would be an interesting assay. Making it commercially available is something that I have no experience in. What we are really working on right now is we're testing other hybrid peptides to see if we can have, this was our winning peptide. At that study, we tested about 16 HIPs. 4 of them had significantly elevated responses, but HIP-11, the one that we find by mass spec in islets and for which we isolate T cells, was our winning peptide. So I think there may be other hybrid peptides there. And once we have a panel of these hybrid peptides, that could be really an interesting target, because you can see we don't get everyone. We only get about a third or 20% of the ones that are there. But yeah, down the road, yeah, that would be great to have.
Well, the Immunology of Diabetes Society would love, and everybody can do ELISpot assays. They're trivial, and if you could come up with a panel of things to screen, it would be a real boon for the field. But then, I always ask you, does putting stress on a beta cell increase the yield of these HIPs? And you've always told me no, but in your talk today, you gave us other clues as to what might regulate HIPs, and that's the stoichiometry of the cathepsins and the products and the kinetics of how long those things are in the granules.
Absolutely.
Have you thought about how that may tie into T1D pathogenesis?
Yes, I have started to develop some thoughts in that direction. So one thing that I'm thinking, as the type 1 diabetic gets fewer and fewer islets, they become more stressed, and the granule will have a shorter half-life because it has to get dumped out all the time to replenish. So the half-life of the granule is shorter. And I told you that incubation time plays a role in driving the formation of hybrid peptides. Potentially, this could mean that hybrid peptide content will go up. So I would like to call this metabolic stress, and I can see that metabolic stress can potentially lead to a higher peptide yield, thereby accelerating the disease onset. However, I'm not sure that that's something that we're starting to work on and we're interested in pursuing.
Well, and then my last question is, have you looked in the I'm sure you have, in the GWAS data to look if there's any correlates with the cathepsin S or cathepsin D and L in T1D risk?
I have not looked, but I think I should. Yeah, thank you for pointing that out. Yeah.
A question. So you mentioned like stress and so on. So is this then something, a process where you're developing these hybrid peptides as part of a stage after the initiation of the disease process, or is this the initiation of the disease process?
So I think the hybrid peptides form, period, in healthy and diabetics. And so far, we've only used mass spectrometry in human islets of organ donors that did not have diabetes. And we did find hybrid peptides in there. It's a regular biochemical reaction that takes place in the granules. However, in type 1 diabetes, you have a certain HLA, and the HLA, depending on the type you have, binds the peptides differently. So I think in the at-risk individual, the hybrid peptide gets presented at its junction, making it most different. However, in non-risk HLAs, the hybrid peptide junction may not even get presented. Instead, it presents either the insulin peptide or the other peptide, and therefore does not lead to the target as a target for autoreactive T cells. So I think that's what's going on here. And we've seen so far all the HIP-reactive T cells that we find present the hybrid peptides right at the middle of the core. The left side is insulin, the right side is the natural cleavage product. So I think that's what's going on here, the HLA, which is the highest risk factor for getting type 1 diabetes.
All right. For those who think I might have dozed off and went to sleep. There isn't a second talk today. Michael Stitzel had to opt out at the last minute due to an urgent matter. I want to be sure that everybody knows that we have 3 sessions scheduled for 2022. The first Wednesday, 2 p.m., January 5, February 2, March 3. And the next session, Ebony Carter, Jonathan Flack on January 5, Zach Knight on February 2nd. And we're hoping to slot Michael Stitzel into that slot as well. And then John Campbell and Daniel Saradini on March 3rd. Kylie is going to send that 2022 schedule to you as an attachment on the calendar invite going forward. But for today, that was a scintillating talk. That was really interesting, very different, and really exciting. Really enjoyed that. Congratulations and good luck with your paper. We'll all be looking for it.
All right, good. Thank you so much. It was fun.
All right, good work. Thank you,
Thank you
everyone.
for great questions, everybody.
Great job, Tom. Thank you. And great work.
Thank you, Dave.